Okay, so this video will go over DNA replication as well as transcription and translation, and a little bit on genetic modification or genetically modified organisms. So when DNA replicates, the double helix has to be unwound. This is carried out by an enzyme called helicase. A series of other enzymes will then attach and essentially will bring in the complementary base so that two new strands of DNA can be formed, and eventually you'll have two identical DNA molecules to the original strand. So again, because of the way DNA works, each base only pairs for one other base. So if originally this T was with this A before they were unwound by helicase, afterwards this A, when you form a new strand, the enzyme is going to only stick a T there because A always goes with T in DNA. And this C was originally bound to this G, so in the new strand that's forming, it's always going to get another G. So the same bases that were in the original DNA will have to end up in the new strand because each base only fits with one other base with those hydrogen bonds. So what you end up getting is that as these things are unwound and then copied by DNA polymerases, those are the enzymes that do the anabolic process of copying the DNA, you get two new exact copies of the original because they only bind to those one other base for the nitrogenous bases. Okay, transcription and translation, though, is a bit more complicated. So yeah, it's great that we have this complicated long molecule of base pairs, but how exactly does that hold our genetic information, and how does that get turned into all the parts of the human body or any animal's body? So the way that it works is that those four bases act like a code. They act like a code with only 4 letters. So it's as if the English language only had 4 letters, and then you were able to write all the different words and get across all the information about everything with just those 4 letters by the exact order that you put them in. So it's a complex idea, but that's really about how it works. The way that we then express the information that's inside of the DNA is that the DNA code is actually coding for different amino acids, and those amino acids get linked up to make proteins. And then proteins are are structural pieces, but more importantly, they are the enzymes. They are the anabolic and catabolic tools that are going to construct and build and chop up and put together the organism. So DNA is sort of the coded information, and then that actually gets translated into things that can actually build and construct organisms. So it's sort of like if you had a gigantic encyclopedia that was written in this code of 4 letters, and then you'd have different sections of that book that would have like different chapters that would tell you how to build things, and what the code had written in letters could then get translated into actually constructing something. So maybe it's a giant IKEA manual that tells you how to build every single IKEA piece of furniture. So while it starts off as just letters on a page, those letters then get translated into actually building, a bed or building a chair. So in this case, we've got this code of letters of nitrogenous bases, but then as that gets read, it gets turned into proteins, and the proteins are the ones that then go out and build everything and work on everything in the body. So DNA itself is stuck in the nucleus. It's got that A, T, C, and G code, the 4-letter code. So the way that the message actually gets out is that it has to be copied down or transcribed, which is why the process is called transcription, by RNA, specifically messenger RNA. But for the chemistry exam, you don't need to know the difference between M and T RNA. So you've got RNA, which is going to copy down the complementary strand of the strand of DNA that it's copying. You're going to get, so if it was A, T, C, and G, the RNA would end up being U, A, G, and C. So it would be that complementary 4-letter code. So you get a strand of mRNA. That mRNA can leave the nucleus, go to the cytoplasm, and end up at the ribosomes. Ribosomes are small organelles where protein synthesis takes place. So that's where the the sequence of bases gets read, and each 3-base sequence on the RNA codes for a specific amino acid, one of the 20 amino acids. So at the ribosome, the different amino acids, the correct ones get brought into place, and they get bound together through condensation reactions, and then you formed proteins. So DNA, you've got this messenger RNA that copies. After it copies the complementary strands, it then is leaving here, where it goes out to the ribosome. That's the blue thing is the ribosome, and these other things called tRNAs bring in the correct amino acid that matches for these 3 bases. So then the next one's going to bring in the bases that match here, and so on. So in transcription, a specific section of the DNA is unwound, again, by an enzyme like helicase, and the single strand of mRNA is copied. And so you can give yourself a try. Can you come up with the correct mRNA strand from this lettered sequence of DNA? It should be pretty simple. So it's just a single strand. It copies just like another strand of DNA would, except there are U's instead of T's. So that's like the main difference. Okay. Then it can leave. It goes out into the cytoplasm to the ribosomes, and this is where it gets translated into the language of proteins, into the language of amino acids. So every 3 letters, every 3 bases codes for a specific amino acid. So once the once you hit the right 3-letter code, you begin a chain of amino acids, and then each 3 letters is going to code for the next amino acid. And so the right amino acid always gets brought in. Because if this is AAA, that's going to bind up with this other kind of RNA that has UUU, and UUU always has lysine bound to it. CAC always has valine bound to it. These are the amino acids. So every little 3-base sequence on the mRNA codes for a specific amino acid, and those little triplet-base sequences are called codons. This stuff that's bringing in the amino acids has the anticodon. It's the exact complementary 3-base strand, and they bring in the amino acids to get condensed together. And that's how you form a polypeptide chain. It's how you form a protein. And that will go on until you hit a certain sequence of bases that means stop. There are a couple of different stop sequences, and that's when the chain will stop being put together. It will then be released. Maybe it's ready to do its job. Maybe it needs to be further modified, but you've assembled the chain of amino acids. And so with a 4-base code, there's actually 64 different triplet sequences that are possible, but there's only 20 amino acids to code for. So a lot of amino acids have more than one triplet code that codes for them. Like you can see here, serine has 4 different codes, all of which code for serine. Most of them have more than one. Some of them, I believe, only have one. Methionine only has one, AUG, the start one. Okay, but you can see end, the ones that say end up here. So there's 3 different ways that if this triplet code gets read, that tells the ribosome to stop making the polypeptide chain. And so then the polypeptide chain gets released into the cytoplasm for further modification or for its time to function. So these are the ones that stop the synthesis of the protein. So here's some practice questions to give a try to. This one is working forwards. So if this was the DNA sequence, figure out the mRNA that binds to that, and then figure out the chain of amino acids that's going to form. And in this one, you're working backwards. If you can see that these are the amino acids that are all in line. So what must the anticodons be? What must the codons be? And then therefore, what must the original DNA be? So the final thing about DNA or nucleic acids to know for the chemistry exam has to do with genetic engineering and genetically modified organisms. One of the most amazing things about DNA is that every living thing on the planet, whether it's a tiny little bacteria or it's you or I, our genetic information is stored with the exact same kind of molecule. We all use DNA and we all use RNA to code our information. That's pretty amazing. It's like if every single book on the planet was recorded in the exact same language with the same 4 letters. It's pretty darn cool. Now, what that does mean is that if you chop out a piece of something's DNA, you can actually fit it into another organism's DNA, and it still works because it's all the same kind of molecule. It's all phosphate backbone. It's all the same four nitrogenous bases. So if you can splice in different sections of DNA, you can then code for different proteins. So, like if there's a particular protein that makes something glow, then if you find the sequence, the gene, the part of the DNA that makes the organism glow, you can then cut that bit of DNA out. You can figure out a way to splice it into another organism, and now they can make a protein that glows inside of them, because in each case, it's just DNA being read, mRNA being copied, and then proteins being built from that original recipe. So it'd be like ripping a page out of one book, recipe book, and gluing it into the other, and then a person reading through that recipe book could then go ahead and follow that recipe and build something, even though it wasn't originally in that book. And so that process is known as genetic engineering, and the organisms that result are known as genetically modified organisms. Mainly where we're going to think about this for the chemistry exam has to do with genetically modified foods. There's a list of sort of pros and cons. Benefits include that they can boost resistance to disease and pests like insects. They can make crops tougher against drought and other rough conditions. They can grow bigger, they can grow faster, they can give higher yields, they can have higher vitamin contents, they can ripen more slowly if that's what you desire, so they last longer. So there's lots of positives about genetically modified foods, but the concerns about them are that the long-term effects aren't known because they haven't been around that long, that they could lead to issues in the ecosystem because as plants cross-pollinate or as things trade around their DNA, you could pass along resistance to all sorts of pests, which could lead to terrible consequences in the ecosystem. They could be linked to allergies, and another issue that's not really what would they do to the environment, but just people are concerned about them because of a lack of transparency in food labeling in some countries. Like people don't know whether or not their food is genetically modified or not. So that's just a tiny little tangent that the IB has added onto this topic, but it connects with the idea that we're all made of this same stuff, this same DNA. So if I chop out a piece of my DNA and stick in the DNA from something else, my body can still read it and it can still make proteins from it. And here's the checklist, and that wraps up nucleic acids.